conflicts of interest, and other biases in research. Regina Nuzzo's How Scientists Fool Themselves and How They Can Stop, with discussion questions connecting the Sujin et al. Paper, The Ethics of Characterizing Difference, Guiding Principles on Using Racial Categories in Human Genetics. This 2015 article, written by Dr. Regina Nuzzo, who's currently an American Sign Language professor at Gallaudet University, examines why many scientific findings fail to replicate, which is a problem often referred to as a reproducibility crisis. So Nuzzo argues that the issue is not usually fraud or intentional misconduct, but rather the result of common research practices combined with cognitive biases. The reproducibility crisis refers to the growing recognition that many scientist studies cannot be replicated when other researchers try to repeat the same experiences or analyses. In other words, results that were originally reported as significant often fail to produce the same findings when the study is conducted again. And this problem can be seen in several fields: psychology, medicine, biology. To understand if there really is a reproducibility crisis, I turned to this 2016 study where they found that they tested 115 scientists to really understand, is this a thing that's happening within the community? And they found that across different scientific fields, scientists were having a difficult time reproducing their own scientific research as well as others. And for Nuzzo, this might not mean that all science is broken, but that the system may systematically favor results that look stronger than they really are.
A team of physicists reported a startling discovery. Neutrinos traveled faster than the speed of light by 60 billionths of a second in their 730-kilometer trip from Geneva to a detector in Italy. Despite 6 months of double checking, the bizarre discovery refused to yield. But rather than celebrating a physics revolution, the researchers published a cautious paper arguing for continued research in an effort to explain the observed anomaly. In time, the error was tracked to a single incorrectly connected fiber optic cable. This example reminds us that real science is more than static textbooks. Instead, researchers around the world are continuously publishing their latest discoveries, with each paper adding to the scientific conversation. Published studies can motivate future research, inspire new products, and inform government policy. So it's important that we have confidence in the published results. If their conclusions are wrong, we risk time, resources, and even our health.
For Nuzzo, scientists fool themselves not because they are dishonest, but because human cognition is biased and the structure of modern science amplifies those biases. Scientists are often working with large data sets and flexible statistical methods, which allow many possible ways to analyze the data. But because researchers are also human, they may unknowingly favor results that confirm their unconscious bias. According to the Oxford Review, unconscious bias, also known as implicit bias, stems from our brain's tendency to categorize and make quick judgments based on past experiences, societal stereotypes, and cultural norms. These biases operate without our conscious awareness, influencing how we perceive, evaluate, and interact with others. While they may not reflect our true beliefs, they can significantly influence our attitudes and actions. At the end of the day, Nuzzo is saying our brains evolved for survival and not statistical reasoning. It is obvious that our statistical methods have barely caught up, that our institutional norms and statistical conventions still operate as if we are running small, controlled experiments. Yet we have huge data sets, many variables, and countless possible comparisons. More data feels like more truth. There's a high output of research, and there's a lot of publication pressure on scientists. So Nuzzo identifies several cognitive traps, as she calls it, that contribute to these problems, including hypothesis myopia, Texas sharpshooter, asymmetric attention, and just so storytelling. Hypothesis myopia refers to a tunnel vision around a favored hypothesis. So instead of asking which explanation best fits the evidence, researchers will often ask, can I find evidence that supports my hypothesis? The problem is that confirmation feels like proof, even when alternative explanations were never seriously tested. scientists may unconsciously design studies or interpret ambiguous results in ways that favor what they already expect to find. The next cognitive trap, a Texas sharpshooter, where a researcher examines this large data set, notices a cluster or statistically significant results, and then treats that cluster as if it were the original target. The pattern is identified first, and then the hypothesis is constructed afterwards. The bullseye is drawn around the data when something interesting appears. And the brain is wired to detect coherence and similarity, so the cluster feels real and intentional.
Disco record sales were up 400% for the year ending 1976. If these trends continue, hey!
Your fish are dead.
Yeah, I know. I can't get them out of there.
The next trap, asymmetric attention. People are often criticizing or scrutinizing very surprising results more carefully than the expected ones. So if your data aligns, they're accepted. And if the data somehow contradicts, researchers are often looking for errors or alternative explanations. This creates a disconfirmation bias, so that evidence fits prior beliefs receive less critical examination than evidence that challenges them. And so my personal favorite cognitive trap, the just so storytelling, refers to this tendency to create a neat, convincing explanation for a finding after the result is already known. And this name comes from Rudyard Kipling, Just So stories, which is how the leopard got its spots, which are satisfying stories, but they're actually invented after the fact. And in scientists, this happens when a statistically significant result appears. Researchers then construct this theoretical explanation that makes the results seem expected or inevitable. That explanation is presented as though it motivated the study. The danger is not the lying, but the danger is this narrative coherence. The story makes the evidence feel stronger than it actually is. To address these issues, Nuzzo suggests several reforms aimed at improving scientific reliability. These include greater transparency, which means making data, methods, and analytic decisions visible to others, a team of rivals where researchers with opposing theories or interpretation actually work together to design a study before the data is collected. And because each side helps determine the methods in advance, it should help reduce this confirmation bias. There's also blind data analysis, where researchers analyze data without having known which experimental group is which or which condition is expected to produce a certain outcome. This potentially prevents expectations from subtly influencing analytic decisions by temporarily hiding outcome labels. Researchers reduce the risk of steering results towards what they unconsciously expect to find. And so the other paper that we read this week, the Shujin et al. Paper, The Ethics of Characterizing Difference, Guiding Principles on Using Racial Categories in Human Genetics, actually flips the concern that Nuzzo has, where instead of focusing on our methodological practices and these cognitive biases, the Shujin et al. Paper asks us what are our ethical responsibilities as scientists, especially when studying race and genetics. And so while we might understand race to be socially constructed, that seems to vary in these very biomedical genetic studies texts. Poorly framed genetic research can unintentionally reinforce racial stereotypes, biological essentialism, or racial hierarchies. And then these interpretations can contribute to discrimination or harmful social narratives. So for biological essentialism, we're treating race as a genetic factor rather than a social construct. We're misinterpreting research, so media or public may actually exaggerate racial genetic differences. There's a reinforcement of social inequalities, findings that support existing racial hierarchies, and then the conceptual confusion. What is race? What is ancestry? And then the population genetics often get conflated. And in this open letter by Shujin and their colleagues, the authors tell us 10 guiding statements or principles about how racial ethnic categories should be used in genetic research. Now, these are not rules per se in a strict sense, but these kind of normative guidelines or ethical descriptions of how a scientist should act and in order to prevent misinterpretation of genetic variation. So we'll see something like race is historically and socially constructed. Genetic variation does exist, but it does not map neatly onto racial categories. Most genetic variation occurs within populations rather than between them. This means there's only a small portion of genetic differences among humans. Researchers must be cautious when using race as a proxy for genetic ancestry. There needs to be clear definitions of racial and ethnic categories. Researchers should distinguish between race and ethnicity and ancestry. Scientists should recognize the social and ethical consequences of research on race and genetics. There needs to be interdisciplinary collaboration. Communications of research finding must be responsible and careful. And at the end of the day, research on human genetic variation should aim to improve health and scientific understanding without reinforcing racial inequality or discrimination. And so 2 questions I thought we could think about at home is, Newzone warns us about just so storytelling, where researchers create explanations after seeing results. How might this type of storytelling actually influence explanations of genetic differences between racial groups? I also wanted to ask, both articles discuss ways that scientific interpretation can shape public understanding. What responsibilities do scientists have when communicating research findings about race, genetics, and human difference?